So, good evening, everyone. On behalf of Kaustav's Careerica, it gives me immense pleasure to welcome you all to today's expert lecture on biotechnology in personalized medicine, being conducted as a part of Module 2, Translational Biotechnology and Healthcare Innovation under the Winter Internship Program. I, Yash Nigam, Ambassador at Kaustav's Careerica, pursuing my Master's in Biomedical Sciences from Dr. B.R. Ambedkar Center for Biomedical Research, ACBR, University of Delhi. Due to some urgent work, our founder, Dr. Pratima Tripathi, is not able to join us today in this lecture. So, on behalf of her and Kaustav Careerica, I welcome you again to this expert lecture. To begin with, let me briefly introduce Kaustav Careerica, which is an initiative under Prakrit EdTech Private Limited, founded by the Dr. Pratima Tripathi, Director and CEO, Kaustav Careerica. So, the Careerica established with a clear vision of bridging the gap between the academic knowledge and real-world application in the life sciences, biotechnology, and healthcare sectors. The vision of Kaustav's Careerica is to create a future-ready scientific workforce by integrating education, research, innovation, and career mentorship. Our mission is to empower students and early career professionals with structured learning pathways, expert guidance, and hands-on exposure that enable the informed academic and professional decisions. Our services include internship programs, expert lecture series, research mentorship, skill development workshops, academic guidance, and career counseling, which is all curated to align with the evolving industry and research demands. Over time, Kaustav Careerica has successfully reached learners across diverse institutions, delivered the internship programs, and built a strong network of academicians, clinicians, researchers, and industry experts. Our long-term goal is to nurture not just degree holders, but competent professionals, innovators, and responsible contributors to the healthcare and society. This winter internship program is one of our flagship initiatives designed to provide students with exposure to interdisciplinary and translational domains that define modern science and the healthcare. This program is structured into the focused modules, each addressing critical areas such as diagnostics, therapeutics, personalized medicine, innovation, and entrepreneurship. The Module 2, Translational Biotechnology and Healthcare Innovation, focuses on how the discoveries made at the laboratory bench are translated into the clinical applications, patient-centric solutions, and healthcare technologies. The personalized medicine stands at the heart of this translation, where biotechnology enables the precise diagnosis, targeted therapy, and improved patient outcomes. Today's lecture is therefore extremely relevant as it explores how biotechnology is reshaping personalized healthcare through the molecular insights, clinical research, and precision-based interventions. It is now my great honor to introduce our esteemed expert speaker for today's session, Dr. Ishan Pandey. Dr. Pandey is a highly accomplished medical microbiologist and translational research with the extensive experience in the clinical research, molecular microbiology, oncology-associated infections, and personalized medicine. He completed his MSc and PhD in medical microbiology from the Department of Pathology, Motilal Nehru Medical College, Prayagraj, where his doctoral research focused on a clinical trial-based study investigating the Helicobacter pylori and its role in the gastric cancer, which is a topic of immense relevance to the personalized diagnostics and targeted therapy. Currently, Dr. Pandey serves as a senior research scientist at the Department of Health Research-sponsored Multidisciplinary Research Unit (MRU) at the Motilal Nehru Medical College, Prayagraj. In addition, he is a distinguished adjunct faculty at the Center for Global Health Research, Savitha Medical College, Chennai, reflecting his strong academic and collaborative footprint across institutions. Dr. Pandey also brings with him nearly 9 years of teaching experience, having served in the departments of microbiology and biotechnology at the reputed institutions, including Motilal Nehru Medical College, Prayagraj, AKS University, Satna, and Babu Banarsidass Dental College, Lucknow. His teaching experience complements his research expertise, making him a mentor who understands both clinical relevance and academic rigor. His research journey includes collaborations with some of the most prestigious institutes in the country and abroad. His doctoral work was conducted in collaboration with the Department of Medical Gastroenterology at Motilal Nehru Medical College, the only super specialty wing among medical colleges in the Uttar Pradesh. He has also worked at the WHO National Polio Surveillance Laboratory at SGPGI Lucknow, received training at Jalma ICMR Agra, and served as a research assistant in the Indo-Europe Joint Research Project on HPV in head and neck cancer, involving the collaboration with 16 European countries, including IARC France. Dr. Pandey's scholarly contributions include 20 plus research publications and presentations with a cumulative impact factor exceeding 50, including 2 high impact publications as a first author. His work has been published in the leading journals such as Trends in the Food Science and Technology. He has over 570 Google Scholar citations with an H index of 12 and I10 index of 12, highlighting the impact and consistency of his research. He is an active member of prestigious professional bodies including ASM USA, ACS USA, and the Microbiology Society UK, and serves as a reviewer for several high-impact international journals such as BMC Genomics, Scientific Reports, Frontiers in Bioengineering and Biotechnology, International Journal of Surgery, and Diabetes and Metabolic Syndrome, Clinical Research and Reviews, among others. Dr. Pandey is also a sought-after speaker and has delivered invited talks at institutions such as City University, Ludhiana, Ramaiah University, Bengaluru, Parul University, Gujarat, and many more. His continuous involvement in new research projects and academic roles reflect his commitment to the lifelong learning and innovation. With such a rich blend of clinical research, molecular biology, and personalized medicine, Dr. Pandey is exceptionally well suited to guide us through today's topic. Sir, we are truly honored to have you with us today. We look forward to gaining invaluable insights into how biotechnology is enabling personalized approaches in medicine, from molecular diagnostics to the targeted interventions.
Okay,
Welcome,
thank you so
sir.
much. Yeah. Kaustav, I hope I am audible. And
Yeah. Yes, sir, you are audible.
yeah, and sorry for this technical glitch. It happened
It's
due to
fine,
some.
sir. It's fine.
Yeah, so I will be sharing my PPT with you all. I guess you tell me just if it is visible or not. Is it visible?
Yes, sir, it is.
Okay, just a minute. Yeah, let me make it as a. Is it visible in the full screen mode?
No, sir.
Hello?
Hello.
Hello?
Hello. Yes, sir.
Was it visible in the full screen mode?
No, sir. No, sir. No, sir. Not yet.
Just a minute. Just a minute. Why is it not visible? Just a Now, is it visible?
Yes, sir. Now it's visible.
Is my screen visible?
Yes, sir. Yes, sir. It's visible.
It is in full mode, full screen
Yes,
mode.
sir. Yes, sir. Yes, sir. Screen share mode.
Okay, sure. So, thank you to the organizers, and thank you, Costa, for coordinating with me and getting into it. You have given my introduction as well, so I thank the participants as well. So the topic of my talk today is biotechnology in precision medicine tailoring, which actually the topic is very precise, but it has a very deep meaning into it. Now, what happens is, we talk about in the context of precision medicine, then it is tailoring treatments to the individual patient. Now, there is one saying which is quite going on, that there is no pill for all ill. Now, you cannot say that there is only one pill for one ill. You cannot take only one tablet for each and every person, because each and every person is different. He or she is having a definite genetic composition or a proper lifestyle pattern. So each and every cell is behaving differently in the body. That is why now the medicine or a food, whatever we take, should also be very precise, very focused, and very individual centric. So the basis of what is personalized medicine is person to person or individualized medicine. Okay, so with this, there will be two definitions. Then there are two words, because as you see the title as well, biotechnology in precision medicine. or personalized medicine. Then what is personalized? And one is precision medicine. So key definitions include that precision medicine is tailoring medical care based on individual patient characteristics like the genetics, lifestyle, environment, whatever it is, personalized up to the individual level. And what is precision? Precision means precise. Precision means using molecular genetic data to identify specific targets and match patients to the optimal therapies. You might have also heard about Operation Sindoor. It was precision attack. So they identify the hotspots, the key areas where they have to attack, or where likewise what is talking about this precision attack or the Operation Sindoor, in which our armed forces conducted precision attack. Likewise, this precision medicine or the personalized medicine is also working in a specific targeted manner where we don't have to damage the normal physiology or the normal mechanism of our body, and we have to only target those hotspots or parts which are or when you not talk about the parts, we talk about the cellular environment. We have to target those pathways, those molecular mechanisms, those genes or those proteins where we can directly eliminate the disease, or we can inhibit the disease from forming. And these forms a predictive tool for us so that we can gain previously whether if a person is disease free, he will again get that disease, or what is the prognosis of that disease. So there might be quite a there will be few important terms coming through this, so you will be getting accustomed to all those terms as well. Now, what are the core biotechnological tools? As the students which you are listening might talk, maybe masters, bachelors, or PhD student, BTech, MSc, life sciences students. So there may be biotechnological tools. So what is biotechnology? Means the use of techniques or the technologies in the as domain of biology are biotechnological tools. So you might all have heard in your masters, in your bachelors, if you have done that, about next generation sequencing, biomarker, CRISPR-Cas9, gene editing, bioinformatics might be one of your papers or subjects. So these are four major and basic things which is the core of these biotechnological or medical biotechnology or this precision medicine. So first is next generation sequencing. I maybe just a minute. So next generation sequencing is talking about the DNA or the RNA of the genome. And just a minute, just a minute. Yeah. So next generation sequencing is about the whole genome and the exome analysis. Now the genome is about the whole gene and the network of the analysis. That is the whole genome of a particular cell or a particular organism. It is a whole genome. Now the exome analysis. Now there is the exome also which is present, which is also done for analysis. Now what is biomarker discovery? Now biomarker, as it is known, you use marker pen for highlighting something. So likewise these are the biological markers which help us identify a particular disease or a particular syndrome or a particular condition, we say, inside the human body. So that biomarker may be up to the level of genomic, maybe it can be a gene, it can be up to the level of proteomic, maybe it can be up to a protein, or even now the term metabolomic profiling, which is about the metabolites, means the bacteria, the viruses with the good bacteria which is residing inside our body, or what we eat, what we do is the metabolism. What is our metabolism? So when we metabolite, we produce we perform our metabolism, we produce some metabolic molecules or metabolic chemicals. That profiling is also necessary as a biomarker that our metabolism is not doing fine. Then the terms about CRISPR and gene editing. What is CRISPR? CRISPR is like molecular scissors. Now this is a part of gene therapy. These are a therapeutic interventions up to the level of the DNA, where we can edit the DNA like we do in our laptop, in our systems. We edit our literature or our text as per our need, or the spellings are corrected. Likewise, in the gene therapy module, we can now edit the gene by molecular scissors. These scissors will go, they will cut the faulty DNA or the RNA, and especially the DNA, they will cut the DNA or the sequence, they will specific sequences they will cut, and they will add new sequences. But there might be some negatives also, which we'll be discussing in the later on aspect. Now with the advent of informatics and this big data, AI, now this data, there will be a lots of data. So this data integration and predictive modeling with AI is amongst the bioinformatics tool. Now what is bioinformatics? Is this this accumulating this biological data and using the informatic tools or the data base management, how we can segregate this data into different apps or softwares that we can annotate, or we can give them some classification, how to use them. Rather than accumulating or making it a tedious task, we are using uh, technology to integrate these data as bioinformatics tools. Now the next is, no. Next, we move on that to next generation sequencing. What is next generation sequencing? It is a high throughput DNA oblique RNA sequencing. What is sequencing? It is a common term. You know what is a sequence. Means sequence is when we move on to the DNA or to the RNA, we see there is a sequence of nucleotides which is arranged in a gene or in a mRNA something. So that sequencing is known as how it is ATGC, which sequence it is arranged. And those triplet sequence, you know that they form a codon and they code for a protein. So how this sequence of the three codons are maintained, and how they are making the protein, which is the functional unit for any phenotype to occur. So this is how we see sequencing technologies for genomic profiling. Now in this, there is whole genome sequencing. Whole exome sequencing means there is whole genome sequences is complete genetic blueprint or the complete information about the genetic sequence of a particular organism. And when we talk about whole exome sequencing, that is about the protein coding regions only, because there are introns, exons, introns. You might very well know that what are introns and what are exons, the junk DNA. These are all the terms which you all might be familiar. So this exome sequencing is the only protein coding regions, where we sequence which are the regions which are only responsible for the protein coding. Then there are targeted panels or the micro arrays, you can say the micro array, where there in this only the disease specific mutations are detected. Like there are different KRAS mutations, P53 mutations, BRCA mutations. There are different kind of genes which are known to have mutations responsible for a specific disease. They have associations. So for those, we have targeted panels that specifically see the mutation for those targeted genes. Then there is a rapid turnover enabling time taking, means previous this data, this analysis would take 7 to 14 days to 15 days for reporting. Now it is up to 1 day, 24 hours or 48 hours real time data reporting, so that we can immediately react to the patient for any therapy to start, or any therapy to discontinue, or anything to carry on. Now, second, what I wrote was biomarkers. Now it's types and applications. Then there are genomic biomarkers, means the mutations, single nucleotide, SNPs are the single nucleotide polymorphism, means A is replaced by G is replaced by T. So that is a single nucleotide, and that is a point mutation. So mutations are point mutations, inversion, insertion, deletion, these kind of things. Mutations are responsible. They might be at the level of chromosome, they might be at the level of gene, this way. Then there are copy number variations, means the gene can be duplicated, replicated, that is copy number variation. Then on this basis we can predict the drug response as well, that whether this gene is uh, like there are many genes which are drug metabolizing genes, like the cytochrome genes, cytochrome P450. These if these genes mutate and they become mutated type, and from wild type they start to demetabolize the drug, or they don't metabolize the drug, so that drug moiety may get accumulated in the body and may cause toxic effects as well. So this predicting thus drug response also gives rise to a specific field which is known as pharmacogenomics, which is about the genomic profiling, predicting how a drug is responding to the body. Then there is proteomic. Proteomic approach is up to the level of protein, because we are moving from central dogma, one gene, one enzyme, from one gene, one enzyme to one gene, one protein. Now the dogma has changed, and protein is the now of our concern. With that, protein is the functional part of it. It helps us make the body, it helps us to make the skeletal structure, this, and all enzymes or all systems which are working mostly consist of a protein, maybe it antibody, maybe it something enzymes, all are proteins. So protein levels. Then what are the phosphorylation patterns of the treatment stratification, what is, how it is phosphorylating, how it is cleaved, how this protein is forming. So there may be some conformational changes, how this protein is aligning in 3D structure. So these are also up to the level of proteomic level. Then the third term was metabolomic, like the small metabolites revealing disease pathways and drug efficacy. That there are many small metabolites like the short chain fatty acids. When the good bacteria, the lactic acid bed or the firmicutes, which are residing inside our gut microbiome, they ferment the prebiotics and produce short chain fatty acids, small short chain fatty acids. These are the metabolites which confer good probiotic effect and may give better response to colorectal cancer, to gallbladder cancer, to gastric cancer, to other kind of diseases as well, and may confer good effect to our brain as well. So there, you might have heard about the term gut brain axis. So it will also produce good hormones, because our gut is our second brain, and it is the main organ of our body which is responsible for our immune system. Then cancer precision medicine. If we talk about precision medicine or personalized medicine, it is basically taught about or talked about in case of cancer. So the cancer, what about it is that the genomic biomarkers revolutionizing cancer therapy and treatment selection. Now there are biomarkers in blood. You may test cell-free DNA, or you may see circulating tumor cells. You can identify minute up to the level of a nanogram. You can identify the genes or the biomarkers, and on that basis it helps the clinicians to devise the particular drug or the cancer therapy for treatment. Now there are different kind of mutations as well, like the somatic mutations or the germline mutations. Somatic mutations are the tumor specific mutations guiding drug selection, and there are germline mutations as well, which are in the germ cells or the cells for our progeny. Then there are immune biomarkers like the PD-L1, that is programmed death ligand 1 expression, predicting immunotherapy response. This PD-L1 expression, if it is expressed or if it is upregulated or downregulated, this helps to predict in the immunotherapy when we give inhibitors to check the immune response. Then there are microsatellite instability. These are small set of sequences which are micro MSI sequences, microsatellite instability, MSI it is known. These are identifying candidates for checkpoint inhibitors. Identifying candidates for checkpoint inhibitors means checkpoint inhibitors are like gatekeepers. When you enter inside a campus, you have to go from the gate, and at the gate there are gatekeepers or guards which are present there, which check your identity cards and lets you in. So this microsatellite in instabilities are just like identifying the candidates, whether these candidates or these genes or these drugs can cross the immune system, or it may be metabolized by the immune system. Then comes the cancer precision medicine. Part 2 is about the testing. Now, how to test? As I was telling that there are different genes for this therapy. So I have taken three most important studied, most widely studied in case of cancer. First is the HER2 testing, which is the human epidermal growth factor receptor testing. It guides the therapy for breast cancer and gastric cancers major, but HER2 is also studied in other cancers, head and neck cancers and other cancers also. But majorly it is widely studied in breast cancer, and now up to 30% it is also studied in gastric cancers as well. It means the stomach cancer. Now, for as far in the breast cancer, when there is a treatment response or there is a testing, there is three kinds of testings are done. One is the hormone testing, that is the ER/PR, means the estrogen receptor or the progesterone receptor. ER/PR HER2. Three panel of genes are being tested. Then there are EGFR mutation, that is the epidermal growth factor receptor mutations, which predicts tyrosine kinase inhibitors. I was talking. This is immunotherapy response in non-small cell lung carcinoma. So in case of lung carcinoma, this EGFR mutations that helps us predict at which gene or which polymorphism it is causing, that the tyrosine kinase inhibitors may be given or not. If the particular mutation or a particular polymorphism is there in the this EGFR gene, then tyrosine kinase inhibitors are immediately given without any delay. Then there is BRCA, that is BRCA 1 and 2 status. This is for the risk stratification, and for platinum and PARP inhibitors, like sensitivity drugs, because the platinum drugs like the cisplatin and all these drugs are given, and the PARB inhibitors sensitivity is also given. Then what I was telling that about the pharmacogenomics, means those genes which are responsible for metabolizing drugs and giving response, means drug response prediction, how this drug will respond inside a particular body. This is very much important before prescribing a drug to any particular individual. So these are the genetic factors determining individual drug metabolism and its efficacy or the efficiency. Now there are three ways, the major three ways. There are other also, but what I was talking previously also, I said that CYP450, that is cytochrome P450. This gene variants, these affect the drug metabolism and dosing requirements. Means if this gene is faulty, we might be needing more doses to for a response, and those more doses may cause adverse effect inside our body and may be toxic or lethal to our body. Then there are HLA alleles, that is the human leukocyte antigens. So these human leukocyte antigens also predict adverse reactions. Example, abacavir, means this is a acyclovir, or carbamazepine, carbamazepine. These kinds of drugs are also given, and these HLA alleles, means HLA typing is done, which tells us about the leukocytes response towards these drugs. Then there is thiopurine methyltransferase TPMT, guiding 6-mercaptopurine dosing in leukemia. This is for leukemia, means the blood cancer. Then there, I was talking about CRISPR-Cas9, the molecular scissors. This is for gene editing and therapeutic applications. CRISPR and advanced gene editing enabling us curative personalized therapy. Now, with this we have CAR T cell, chimeric antigen receptor T cell therapy, which is CAR T cell therapy. Genetically engineered T cells, means these are the immune cells like the B cells or the T cells. Now this T helper T cells are being engineered and regulated so that it can help in the form of CAR T cell therapy. Now CAR T cell therapy in India, Dr. Rahul Purwar at IIT Bombay, he has significantly worked on this CAR T cell, and he is having one startup as well that is working on CAR T cell therapy. So this is CAR T cell therapy is very much important. Then CRISPR based editing, I was telling that these go and correct those mutations up to the level of molecules, molecular level, and these are known as molecular scissors. But there is one problem with CRISPR-Cas9. CRISPR-Cas9 or the CRISPR editing is that these scissors don't know where not to cut also. So there are some genes which are not faulty, and they may, because some patient get cured with cancer, but they may become deaf, they may become they may lose vision. So other genes can also get trimmed or edited. Now allele specific silencing is targeting pathogenic variants selectively. Now, which are allele, you know what the term allele, and allele, which alleles are switched on, which are working properly, which are expressing or which are not expressing. So these pathogenic, which are causing pathogenic effects, they may get silenced. Now, what are the key drivers of this gene editing or NGS or this precision medicine? The key drivers are multi-omics integration, AI predictive modeling, real-time molecular monitoring. What this is helping is multi-omics, means genomics, proteomics, transcriptomics. We are integrating all these metabolomics. We are integrating or combining all these multi-omics integration. Then there is AI predictive by the use of AI or the artificial intelligence based CNN, RNN architecture. We are just predicting as a predictive model. We are just modeling up to the level of data, how we can predict, and real time monitoring about the data, about how this disease, how the data is changing in the real time context of a particular disease. Then introduction of AI, introduction to biomedical AI and ML is that AI and when, since AI is much of a talk, I have put a separate session about AI as well. That artificial intelligence and machine learning brings innovation in diagnostics, genomics, and personalized medicine. These assist in disease prediction, drug discovery, and pattern recognition. Now, the major part which is about this AI is pattern recognition. They recognize a pattern which is being followed, and on that basis it gets trained and it predicts and gives you a results. So what happens is when you talk to chat GPT or you talk to perplexity or you talk to any of these generative AI which generate, they also predict, they also get a pattern about which type of questions you are asking, and on that basis whatever answers or whatever data you feed, if you change the question, they will take the previous data and report you on that basis as well. So it will integrate with the omics. Now there is image processing as well, because in medical sector there are lots of images, CT scans, X-rays, slides, these pathological slides, or different kind of ultrasounds, all have imaging. So this image processing is also one of the major area, and then there is clinical data. So there are different kind of tools as well, that if we talk about bioinformatics or if we talk about artificial intelligence, so there are tools like the crime and cell profiler. What is the crime is the visual workflow based data analytics tool. This crime uses gene expression performs gene expression, biomarker dissection, integrative data analysis as well. This is a visual, you can put your data inside it and it will give you the data. Then there are cell profiler. Cell profiler is about the profiling of the cell. It quantifies biological images, used image based cell profiling, phenotypic screening. If you give an image of a diagnosis, it will give you the cell-based analysis and phenotypic screening as well. Then there, this what I was talking about. There are three kinds of basic AI system, which is ANN is basic model for disease prediction. That is the artificial neural network. Then there is CNN, which is the convoluted neural network, which is applied in medical image analysis like the X-ray, MRI, histopathology data. And then there is RNN, which is the RNN, which is suited for time series data like the ECG, genomic sequences as well. This is, we will not go into the main detail of these bioengineering tools. Now we are using CNN to case study and demonstration are like that. We are using CNN to classify histological slides, like this is well differentiated carcinoma, the TNM staging. This is good poorly differentiated carcinoma. This is moderately differentiated this is carcinoma. This is nucleated, and this is tumor. This is malignant. This is, so we use CNN to classify histology slides. Then AlphaFold. AlphaFold is one software for prediction of structure prediction and drug designing. Then AlphaFold uses the structure to predict about the drug, its activity. This is just like a quasar study, that is quantitative structure activity relationship. Then crime workflow demo about the expression classification pipeline, cell profiler after that detecting morphological changes in treated cell. Then I have published one publication which you will be getting to see, that I have taken that table from my data, which gives you the artificial intelligence technologies empowering molecular diagnostic assisted in gastrointestinal oncology. So these are in the left you can see, the topmost left you can see that there are the molecular techniques used in gastric cancer. These are the molecular techniques how we use. Then there are the artificial intelligence based technologies which can supplement these molecular techniques. And what is the purpose of AI in gastric cancer, and the references. So you can see that there is fish, that is fluorescence in situ hybridization, which is a microscopy or a molecular technique to probe based, which can see the in situ hybridization of a gene which has overexpressed, or that copy number variation has changed. So for that, the tool artificial bed tools is Gelfish, GELFSI. What is the purpose of AI is that it improves the accuracy of fish results. Then there is immunohistochemistry, and then there is software which is immunomembrane, so accuracy of immunohistochemistry result. Then there is slide imaging, and the whole slide imaging, or the Fiji or image J is used for microscopy and histopathology. Then there is immunohistochemistry, IHC profiler is there, IHC fish, RT-PCR, Western blotting, we have CNN network. Then slide imaging, we have QPath, the whole slide imaging, image J, plugin as a slide J, microscopy, histopathology, above with its classification and detection of HER2 tumor, non-2 necrosis detection. Then there is QPath, Spotland, RNN, CNN, deep generalized multi-instance learning. So these are depending on, as I was telling you about the classification of gastric cancer type: intestinal, diffused, cancer, non-cancer, poorly, well, moderately, Laurent type, intestinal, diffused, mixed. So this can be used. Then there is ResNet, ImageNet, and CNIME. These all are using, they are supporting in uh, fish accuracy and results. Then what are the future direction and challenges? The future direction and challenges are about the data integration and the regulatory pathways, means about the validation of the biomarkers for clinical use. Now you cannot use all the biomarkers for clinical use. You cannot use all the data for assessment. So up there are 3 or 4 phases of your study. One is the preclinical, then is then you go to in vivo, and then you go to the clinical trials. And in the clinical trials also there is phase 1, phase 2, phase 3, phase 4. So in this you have to first develop something, and then you have to validate in different phases of clinical trial. So once that is validated, it is CDSU approved or it is FDA approved, then only you can validate it as a biomarker for clinical use. Now how this validation is done, it is about connecting the genomic, the phenotypic, and the clinical data together to form a data regulatory pathway for use. Now the most important, which is represented in the green in the second point, is the ethical consideration. Now ethics is very much important, because you cannot challenge the integrity, or you cannot challenge the uh, problem with the human participant. You have to take consent, you have to get him volunteered, you have to fill all the case report forms. These, the privacy of the person, the genetic discrimination you cannot do on the basis of gender as well. You can for the equitable access, you cannot bribe him or her. So there are lots of ethical issues. There are ethics committees, there are human ethics committees, animal ethics committees. These all are formed by the ICMR or by the CDSCO, IPCs. They look very closely into the data. They look very closely into your project before it is being given. Then AI and machine learning is improving the productivity and accuracy of the system as well. Then it is about the, what is the most important use of this genomics or this AI integration is the cost reduction. As I was telling you, that if you get free softwares on your laptop, then what is the use of performing high-end molecular tests which cost lots of reagents, lots of time, lots of space, lots of high-end machinery? So this will improve the cost reduction, making personalized medicine accessible globally. So this is my brief talk, and sorry for that part in which I was not available due to some technical glitch. I hope I was very short and I covered the topic up to the level of the students. So
Thank you, sir. Thank you. So, if you have any doubt, please ask. Else, I will ask sir to ask some questions to you. Sir, I want to share something that here most of the students are from the biotechnology background, and they are the bachelor students. Either some are from the first year and some are in the third year of the program. So there must be some terms and which they don't get right now, but once they will read this presentation again, they will go through this. So if they ever ask some questions, so we will forward to you via email. Is it okay, sir?
hello.
Hello, yes, sir. You got disconnected.
Yeah. Hello. Yeah. Uh, is there a problem with all the participants? They didn't get anything, or they got anything and they are not interested in asking anything?
So maybe they are hesitant to ask
Yeah.
question. So I want to share something that
Yeah.
most of the students are from the bachelor,
Okay.
bachelor program, and most of them are from first years. So
Okay.
there may be
Okay.
some terminologies
Mm-hmm.
which they don't get. So
Yeah. Yeah.
once they
Yeah.
will go through the your presentation again,
Yeah. Yeah.
if they ask, I will forward to you via email.
Yeah. Yeah. Sure. 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 Yeah. Sure. Yeah.
All right, sir.
I
So
hope it
let
was
me.
okay. Okay at your level. You got an insight. It
Yes, sir.
was.
Yes, sir. I'm a master student, so I get
Yeah.
all of your
Yeah.
points.
Yeah. Yeah.
All
Yeah.
right, sir.
So was it good? Was it okay at your
Yes, sir.
level?
It was fine. It was fine.
Yeah. Yeah. Okay. Okay. Okay.
So
Yes.
let me conclude the session with the formal
Yeah.
vote of thanks.
Yeah.
So as we move towards the conclusion of the session, I would like to propose a vote of thanks. I extend my sincere gratitude to Dr. Ishan Pandey for delivering such an insightful and thought-provoking lecture. Thank you, sir, for sharing your vast experience, research journey, and valuable perspectives on the personalized medicine. Your session has really enriched our understanding and inspired our participants. So I would also like to thank the leadership and organizing team of the Kaustav Careerica for their continuous effort in curating meaningful academic platforms like the Winter Internship Program. Finally, I thank all the participants for your active engagement, curiosity, and enthusiasm. So with this, we formally conclude today's expert lecture. Thank you all, and we look forward to your continued participation in the upcoming session. Have a wonderful day ahead. And thank you, sir, for joining us today.
Thank
Thank
you.
you.
Thank you. Thank you.